Good evening, everyone, and welcome to today's talk with James Lundeen. I have the pleasure of speaking with him today, all the way from Cleveland, Ohio. He's unusual. He's a doctor, he's an engineer, he's a chemist, and critically, he's a COVID-19 researcher. So today we have the pleasure of bringing James. How are you today?
Fine, Dr. McMillan. Thank you.
Wonderful. Well, just call me Philip, James. I'll call you James. You call me Philip.
Indeed, Philip.
Good. So, James, just tell us a little bit about yourself, because I think part of what we want to do before we even talk about COVID-19 is we want to know a little bit more about who this person is that we see the picture of and who makes so many valuable posts on LinkedIn. That's the primary purpose.
I was born and raised in Bloomington, Illinois, USA. And by about age 8 or 9, I was repairing tube radios and phonographs. And then I got into repairing lawnmowers and outboard motors and transmissions. And all the time I was taking all the math courses I could take in school. I had a 5th grade math teacher, Miss Pascal. That's a famous name in math, but it's probably no relation. She helped me with some accelerated math projects in 5th grade because the standard stuff was just not enough. Throughout high school, there was a double session, and I took math courses in both sessions, both the morning session and the afternoon session. And I even went on to University High School in summer of 1969 to take algebra trig. And there was an opportunity to work on a teletype computer terminal that was hooked up to one of the defense contractors in St. Louis, Missouri. So we got to spend the afternoons after our morning of algebra trig learning how to program. Mm-hmm. Then I decided to pursue electrical engineering, basically due to my interest in electronics.
No, so then you are a proper scientist. So you are somebody who was, I understand the picture, going into engineering. So how did you transition from that into chemistry and into medicine?
Well, during my electrical engineering at the University of Illinois in Urbana-Champaign, I studied in the Illini Union Hall in the vending room with all of the other nerds around campus. We would set up camp at different tables, and that's where you would leave your things for the day. No one would disturb them. And you had quite a great opportunity to interact with other people who were serious students.
And so in effect, you are openly admitting that you are and were a nerd at that point.
You bet.
You know, if there is anything now, because of COVID-19, nerds are cool. Everybody needs a nerd now, and we need this kind of work to solve this here. So what made you, well, I think you mentioned something at that point with regards to some of your interactions with colleagues. You are a pinball fanatic, aren't you?
Well, I was invited by some of the other nerds in the vending room. We called it the V room. To play pins, and I thought it was the bowling alley downstairs, but when we headed downstairs, it was the pinball gallery. And at the pinball machines, we would carry on rather intense discussions of mathematics and engineering and philosophy and quite a number of topics. It was just it was a very good interaction with like kind.
And so then what made you go from engineering into chemistry first? What was that transition?
Well, I've kind of focused in electrical engineering on biomedical. I took the mechanical engineering course 210, which is the toughest one for the mechanical engineering students. I was the only double E in there, electrical engineering student, and I scored 4th out of 89 students. All the rest were mechanical engineers. And that was about transducers and sensors and data processing. So I was looking at biomedical, and then most of my V-room colleagues were headed into professional degrees, and many of them into medicine, and that piqued my interest. And I realized then that I would need to transition into chemistry or biology in order to make that jump.
I see. So the transition into chemistry was primarily so that you could get into medicine.
Correct.
Absolutely fascinating. And when you started medicine, did you find it what you thought it would be? Were you excited by it? Were you surprised by it? Were you disappointed by it?
I was very excited about it and found that there were so many opportunities to advance the science and so many opportunities to learn by doing a careful history and physical. That's 95% of your diagnosis.
Absolutely, absolutely. And so then you went on into practicing medicine for some time. What then made you transition out of medicine into a private sector? Because you are now in business, and you've got a unique software in terms of receptor binding, the Lundeen software, don't you?
I do. The Lundeen software originated as a medical school project at the Chicago Medical School in 1979. I was fortunate enough to get an award for student research for that project on graduation in 1983, and I formed Lundeen Software and developed software for the pharmaceutical world for receptor binding analysis to be able to discern whether or not there was a high and low affinity receptor involved, and if so, what was the ratio of high to low affinity.
Wow. And so this software is still in use today?
It is still in use in some places today, and it was most heavily used until the radioligand binding assays were diminished substantially, probably mainly due to all the low-level radioactive waste that was being accumulated. So they went into other technologies where they didn't have the other technologies were not as applicable to this, but this software is currently being redeveloped to study toxicity of in preclinical phase of new small drug development.
So in your time, though, you were still practicing for a period of time in terms of medicine. Do you miss that practice work?
I do, but I feel that I'm at a point in life where I need to focus my scientific skills and clinical acumen to help tackle some of the problems that we have now and going forward. One of those is that most companies have stopped antibiotic development, and there are superbugs out there. There are going to be more, and I'm looking forward to having a company that will be developing new antibiotics.
Wow. Now that's fascinating, because that is a huge area, and one that I don't think the general public is aware of. The fact that in reality we are coming to the end of the antibiotic era unless we have some huge breakthroughs with new technology or new forms of antibiotics. So that's a big one, but it's very high risk, which is why many of the big companies have drifted away from it.
But I have an edge. And the edge is the software that's being retooled will be able to help determine toxicities in the preclinical stage far better than determining it in phase 2 or phase 3 of FDA trials. Because with a 95% failure rate of new drug development of small molecules, that's worse than a coin toss. So there are an awful lot of things that aren't measured early on that would help researchers decide what their lead molecule is and what some of the other molecules they would have for backup would be. And if a molecule is very good as being an antibiotic, it also has a high tendency to interfere with the cardiac muscle, and the QT interval can be prolonged, resulting in a sudden stoppage of the heart where you really can't resuscitate the person. First it goes into toward the side, and then the heart becomes asystole, and you can't get them back. So if we can stay away from that, then I think we can make some good strides forward in antibiotic development.
Wow. Is there any relevance with what's being developed with that? This is a little bit of a diversion, but how does this correlate in terms of, say, antivirals? Is that a similar principle?
it would be a similar principle, because a number of the antivirals, even amantadine, can interfere with QT, corrected QT interval, and somebody where the QT is already too long, based on their congenital, based on their genetics. If they take a medicine that prolongs that more, then their heart can simply stop. So it could be an antiviral, an antiprozoic, almost any type of medicine.
So that raises another interesting question, because we have recently, I mean, this is relevant to later on, but I can't help asking it now. In terms of ivermectin, one of the big questions is in terms of the dosing that is being used for potentially in vitro versus in vivo. Seem as though they're quite high. Is that your technique a way that we could work out the toxicity, the risk of toxicity?
Yes, I do believe so, for any of these molecules, because you see, these molecules only work through the atoms in the molecule that are charged or potentially charged at various pH levels. Hydrocarbons have very little to do with the interaction of the drug and the receptor. It is far more the unpaired unpaired pi electrons that are unshared that interact at these binding sites. So oxygen, nitrogen, sulfur. Arsenicals used to be used to treat venereal diseases over 100 years ago.
Interesting. Did you just see that question there, James, with regards to, oh, it's not a question, it's a statement. Antibiotic development needs to be a not-for-profit area with taxation-free status for companies. Any thoughts on that?
I believe that we're going to have some change in intellectual property laws to support antibiotic development at the same time that would coincide with keeping costs contained and being fair.
Absolutely. So that's a big area, and one that we have to probably look at in the future. So, go on. So, moving forward, the big area that we need answers for is to do with COVID-19. And this is primarily where I really wanted to talk to you about some of the research that you've been doing. But on a personal note, has it touched you in any way?
Yes, a couple of my friends, a bit older than I, both had thromboembolic events. One had a cerebrovascular stroke, and another one through a pulmonary embolus last Saturday. Both of those gentlemen passed away. It was both due to fighting off COVID.
Oh dear, I'm really sorry to hear that. So what was the trigger for you to start doing COVID-19 research?
When I realized that vaccine development would be a big challenge to do it on a fast scale, and I also realized that I had already done some literature and clinical research looking at the association of Down syndrome and the development of Alzheimer's, both in Down syndrome patients and non-Down syndrome patients, and found that there was a commonality.
And this is what triggered your interest in terms of COVID-19. And so you have been sharing a concept that I want you to, as simply as you can, kind of bring to us with regards to fibrin and fibrinogen. What really is that about?
What that is about, fibrin is the end product of fibrinogen going into the clotting process along with platelets. And all of the diffusion problems in the lungs and all of the coagulation problems associated with COVID-19 have to do with this fibrin deposition, among other things, but that's a substantial part. And one of the reasons for that is there's an extraordinarily high level of fibrinogen when vitamin D3 is deficient. The concentrations of vitamin D3 and fibrinogen are inversely related. And if you have low vitamin D3, you have markedly high fibrinogen levels. And it's really a 2-for-1 situation. If you have your vitamin D3 level in good shape, then in general, unless you have some genetic variant, your fibrinogen level will be in the range of 200 to 400 units, and that'll be fine. However, if your vitamin D3 is below 30, your fibrinogen level may be 700, and that's not good. Vitamin D3 stabilizes mast cells so they don't degranulate and have massive cytokine release, which would go into a cytokine storm or hyperimmune response. And if that occurs and you have all this fibrinogen sitting there, then you have all this raw material for clots. So if you control your vitamin D3, then you're controlling your fibrinogen level, and you wouldn't have this propensity for throwing all these clots and making all of these depositions in the lungs.
That's a really, really fascinating insight, James. I mean, I look at COVID-19 as well, and I'm realizing at what point you're saying. So, okay, so let me try and see if I can not simplify it, make sure I've got it correctly. What you're saying is that the viral infection makes or predisposes people to having the inflammation in the lung, which tends to cause the oxygen depletion because of clots in the little arteries in the lung. This is made much more likely when you have a vitamin D deficiency. Is that about it?
That's absolutely true.
No, that is absolutely fascinating. No, I mean, okay, all right, let's explore this vitamin D a little bit more, because
sure,
vitamin D has been, well, it hasn't been given enough attention. So the first point is, can you explain why the scientific community has bypassed vitamin D in this way?
I believe that I can. Vitamin D, although it's a vital amine, as the other vitamins are, it holds a special place in that it's a steroid hormone. And that's really gone unnoticed for its importance in health. And most people are, if you're a clinician, you're treating, maybe as an endocrinologist, perhaps you're treating diabetes and some other things. If you're a cardiologist, you're treating the blood pressure and the cardiovascular disease. If you're a pulmonologist, you're treating the idiopathic fibrosis.
Mm-hmm.
So you're really looking at your area, and since this hasn't really been brought to the fore, that there's this commonality of a vitamin D3 deficiency as perhaps the starting point, or at least an aggravating factor in development of these comorbid diseases. Since it hasn't been common knowledge, then that wasn't reached for when the COVID problem came on. We've never had a virus in recorded history that's had these types of unusual spectrums of sequela and going from mild all the way to death, or to prolonged post-acute illness syndrome with the long haulers. We haven't experienced this. We didn't have something to compare it with. It's not the same as 1918 at all.
Mm-hmm. Mm-hmm. This is a really, really important point, James. No, I mean, I have looked at vitamin D as well, but I've been looking at it more in relation to how it can trigger certain or increase the risk of certain autoimmune responses. That's one side of it. But what you have said here does seem to be able to basically explain what we see in terms of the lung problem, because the primary issue, other than inflammation, is the fact that the blood is clotting in the lungs, and it's blocking all these microthrombi in the endothelial vessels of the lung. So in effect, you're saying that with vitamin D, even with a severe infection, this clotting is much less likely to happen.
It's much less likely to happen for a couple of reasons. Predominantly because the fibrinogen level won't be high, but also the vitamin D, if it's supplemented or if it's normal, then the mast cells are stabilized and they won't degranulate to any significant extent. Therefore, you probably won't have a massive hyperimmune response with the clotting that would be attended to it.
So here's a question that somebody has asked, and it is quite relevant. Mike has just asked, vitamin D is fat soluble. Do we have a formulation that can bring the levels quite high intravenously as opposed to just orally or even intramuscularly?
Yes, there's one that's to treat the parathyroid disorder that can be used much more quickly. That can bring it up faster. Fibrinogen, the half-life of it is 4 days. So if you were to give an intramuscular injection of 300,000 units of vitamin D3, that's typically being done in some hospital settings with quite a bit of success now. That brings the vitamin D3 level up gradually and maintains a depot reservoir to continue elevating it while the fibrinogen's coming down. So if the fibrinogen, say, started at 700 and your target is 350, well, it'll get there in about 4 days. So it's an acute phase reactant. It also goes up when insulin is released. So after you have a meal, 20 minutes later, your insulin is being released and your fibrinogen shoots up for a bit. It's an acute phase reactant. Now, when you supplement with vitamin D3, while the fibrinogen levels will go down, your other acute phase reactants that are normally measured, CRP, sed rate, ferritin, they don't react or respond at all or appreciably to the administration of vitamin D3. But those are the markers people have been following because measurement of fibrinogen and measurement of vitamin D3 are more complicated laboratory tests.
You've really hit on something here. So, okay, let me just utilize what you're saying here, because vitamin D does seem to be a very simple mechanism where in some estimations we could have probably reduced death by over 75% with just vitamin D supplementation. So that's quite a significant impact that it could have. So there are two ways of looking at it. There is the hospitalization phase where people have already come in, and the fact is that by this point they're already making microthrombi in the vessels of the lung. They're already struggling to breathe. Even when you give them vitamin D, you're right, it will take at least 4 days for the impact to happen. Is there a case for using vitamin D much earlier as a preemptive, almost a prophylactic treatment?
Absolutely. If our diet supported that, or if our sun exposure supported that, we wouldn't have this problem with the COVID-19 as we see it today. But it's not found in many foodstuffs, and there are many different reasons why you won't convert the precursor D2 to D3.
And so, well, here's a good question. How do you explain, therefore, what's happening in, say, Brazil? In Brazil, they clearly don't have a problem with sun exposure, but they are being very, very severely affected by COVID-19. I've been looking at that as well, because I've been thinking maybe Africa is not as severely affected because of the sunlight, but I can't fully explain COVID-19 in Brazil. Any thoughts?
Part of it could be their genetic tree. Part of it could be they don't want to be out in the bright sun. As in Egypt, people don't spend their day out in 40 degrees C weather with bright sunlight. They start their day in the evening. So a lot of places where there's plenty of light, people are avoiding it.
Yes, that could be part of it. Well, they certainly did find in Brazil that the vitamin D levels were low as well, which was surprising, even though they were supposed to have a lot of sunlight. So it still fits within the framework of what you're talking about as being primarily about the microthrombi that are formed in the lung, causing or worsening the disease. So one of the other areas that you'd mentioned was Down syndrome. How does this fit in that picture? Because you've had a focus on that as well.
Yes, the Down syndrome, trisomy 21, with the extra chromosome, it's making more proteins. One of those is the beta amyloid is in a higher concentration throughout the life of a Down syndrome individual. And also they've been found to be chronically low in vitamin D3 in general, although earlier studies said no, let's say 20 or 30 years ago. The more current studies are more realistic.
And so would it explain how the levels or how they increase risk of mortality with regards to Down syndrome? Is that enough to explain it?
I believe that the extra chromosome and the extra protein generation makes their serum more viscous. It's probably a bit more fibrinogen. The fibrinogen is up if their D3 is down, and as I said, more recent studies are showing the chronic D3 deficiency in Down syndrome, as well as in our general population. Earlier studies said they were just fine. They didn't even need 400 units.
And so what would be I mean, you can't give too much recommendation to people, but we can probably give them some general advice. What would you say to your family, with regards to vitamin D, if they want to reduce the risk of severe disease? What would you say to them?
Well, I'd tell them to be sure to take their multiple vitamin with the vitamin D. The Centrum has 1,000 units in it. But if somebody is obese, has type 2 diabetes, they're a smoker, they have other comorbid conditions, in the context of COVID-19, I think they need to take multiples of that 1,000, whether it's 2 or 4 or 5, for a short time to try to get through this pandemic. Because the older you are, the fewer T cells you have, and that's another risk factor. So I found that the T cell depletion is a risk factor. Elevated fibrinogen is a risk factor. Decreased vitamin D3 is a risk factor. Hereditary tendencies is a risk factor. And if you have any overlap of those Venn diagrams, you're substantially at risk. Yeah,
and this probably fits in with regards to what we see in that I was saying to someone recently, and I was trying to figure out a flow chart where sometimes what you see is you see patients that get COVID-19 positive. You anticipate they're so frail that you think to yourself, there's no way they can survive it, but yet they go through it with minor symptoms. Then someone else that you think would be okay gets quite severe symptoms and potentially dies with COVID-19. So I agree with you that there do seem to be a number of factors that have to come together in order to significantly increase that risk. So go ahead, say.
I think some people are more inclined to have cytokine release, even at low levels that aren't really noticed in the milder forms. So they are making the microclots, but perhaps they get through it and fibrinolysis happens afterwards and they don't have too long of a convalescence. Where in other people, the clots are so bad that they have a stroke or an MI and they're gone, and everything in between. It's a real spectrum of inflammation and coagulation.
And so therefore, what are your thoughts with the more recent promotion with ivermectin? Have you any thoughts on that?
I sure do. I looked at that, and some time ago I put up an initial link to this and said that there is a basis for it to be helpful here, and that while it doesn't bring up your D3 level, it lowers your fibrinogen level. So although you don't have the D3 to stabilize your mast cells from cytokine release, you don't have much clotting factor precursor floating around ready to turn into clots. So it's helping half the equation. So you can have situations where the fibrinogen is genetically low or high, or it's up because of the vitamin D3 deficiency or an overlap of these.
Wow. I really like this theory, James. As I said, I've been looking at COVID-19 and there's certain gaps, so to speak. And I do feel that this fills a very, very important hole in some of my thinking, certainly when it comes to the more severe part of the disease. If this is down to fibrinogen and the way how fibrinogen impacts on the blood flow through the lungs. So that's quite fascinating. So moving this along, so we understand the severe disease. Where do you think the long haulers fit?
I think the long haulers fit in that they have in my estimation, they're a long hauler because they survived the acute phase, but the acute phase was most likely brought on by vitamin D3 deficiency and elevated fibrinogen. Therefore, they're probably suffering the after effects of massive fibrin deposition in their lungs, maybe in their arteries. They've had a hypoxic situation, so they may have some organ damage. So they're having a lingering state, and fibrinolysis, or it's also called thrombolysis, is a slow process. It can be complete or incomplete. It can take days, weeks, or months. And the sooner that you get it dissolved, the better. Otherwise, it'll probably calcify and be very difficult to dissolve after that time.
Interesting. Look at this point here and see your thoughts on that. Obesity incidence and deaths per million, they almost all coincide, linked to vitamin D. Any thoughts on that?
Absolutely. I researched whether or not a person's appetite is impacted by vitamin D3 deficiency or insufficiency, and found links to articles showing that if you're vitamin D3 deficient, you have an appetite that's hard to satiate. Therefore, you would tend to become obese, snacking or eating too much, because you're still not replacing your vitamin D3. And then when you get to a certain point, any vitamin D3 you might accumulate is going to be shifted into your adipose tissue and not circulating. And also the decreased vitamin D3 causes insulin resistance. So there's your adult onset type 2 diabetes mellitus originating from an increased insatiable appetite due to vitamin D3 deficiency. I can't explain that. No one ever explained it that way in medical school.
I know. You may not realize this, James. And sometimes when I'm talking to people, I don't have the time to assimilate all that they have said. But what you're saying here is actually tying into quite a bit of the research I've been doing previously with regards to dementia, the links to type 2 diabetes. And I can see how the whole of this is correlating, where in truth, what we are having is a significant number of lifestyle diseases that are related to either specific deficiencies or toxicities in our body, making us prone to more significant disease. And the beauty or the benefit of COVID-19 may be that as we have researchers like yourself focused on this, we may be able to finally tip the balance where people recognize that what you eat is really critical to your health.
Absolutely. And whether or not you use nicotine. Because that raises your fibrinogen levels all by itself.
That is a very, very good point. Really, really good point. And so when we think about the future, therefore, for the long haulers, just coming back to it, what therefore can be done with those people who have persistent symptoms? Is it simply just about vitamin D deficiency, or is it now something else that is prolonging these symptoms?
It's dissolving the fibrin deposits, in my estimation.
And this is where they need vitamin D. And so Priyal has asked, Denny has asked a question here about the latitude factors and solar radiation. And they've found vitamin D sensitivity in many immune, autoimmune, and cardiovascular diseases as well. That probably is quite true. That's quite true. And Priyal had raised this point here as well about pharma companies having a vested interest in keeping the public in the dark about the impact of vitamin D. Any thought on that?
Not going to step on that landmine. I don't know the answer, and I don't want to know their intentions. Nothing implied or inferred. No.
Yeah, I completely understand. So then when we look forward in terms of where we are now, what do you think our answer is going to be? Is it going to be just about vaccination? Is it going to be a combination of strategies? What are your thoughts as to the future with regards to COVID-19?
Well, let me break it down into categories there. First of all, I support vaccinations that are safe, effective, and enduring. I realized early last year that this was a new process of vaccine development in a short period of time would be a Herculean effort, and thought, well, we need to do something as a collateral process of therapeutics that we have. The old adage, going to war with the army that you have, not the one that you want. Also, the best example of a short duration success in vaccine development is the mumps vaccine. That took 4 full years. And the mumps wasn't mutating all over the place. We know the cold viruses mutate all the time.
So let me put you suddenly now in charge. So you now, James, have been brought to the forefront of making the decision long term, not just for the USA, for the world. So you're in charge of a global health organization, and they're saying to you, James, how do we move forward? What are you going to tell them?
You have to use whatever supportive measures you have now, and you really have to find a way to synthesize vitamin D3 and not get it from animals or fish, or from the lanolin from sheep, because some people have an allergy to wool, and the D3 derived from lanolin from sheep wouldn't be appropriate, and they wouldn't know ahead of time.
Explain, or just pause there a minute. You just said something there that caught my attention. What do you mean in terms of the vitamin D from sheep?
Well, sheep have a very thick coat of sort of an oily substance that can be harvested when they're sheared, and they get the lanolin out of that, and they can get the D3, because animals make D3 in their fur, not in the skin, because they don't have skin exposure.
Wow, didn't realize that. Right. I think that this here question from Alicia was about the amount of vitamin D. I think we'd mentioned that already. You recommend at least 1,000 a day, but depending on your risk factors.
If your risk factors are there, or your exposure, let's say that you're a nurse or a doctor, and you're at the front line and you're overweight and diabetic, you have no business being there, number one. But if you're going to be there, probably 5,000 to 8,000 a day for the frontline people with comorbid conditions. Seriously.
And then he has asked, what about your suggestions on raising vitamin D in people who don't respond to supplements?
Who don't respond to supplements. There are other forms of vitamin D3 that are synthetic analogs, but we don't have a lot of data on that. But that's certainly something to look at.
Yes, a very, very good point. Yes, so wow, thank you very much, James. This has been absolutely fascinating. So I'd like you to close out in terms of your prediction for the future, where you think that we probably should go as a society, and what learning should we gain from this very, very difficult experience with COVID-19.
Thank you, Philip. I sincerely believe that we need to supplement the vitamin D3 levels in people who are at risk in particular, and then to get to a point with laboratory testing where vitamin D3 and fibrinogen testing is straightforward, simple, and can be done at high volume. Right now, they're more laborious types of tests. That's why other acute phase reactions are CRP, ESR, ferritin are used instead, because they're simpler to perform and perhaps more readily available. So we have to have people thinking in terms of good health and preventative medicine, and doing away with the nicotine use in any form, because in any form it raises fibrinogen levels. And it's going to be difficult from many points of view, because it's going to be a change in lifestyle and a change in diet. We also have to find a way to synthesize vitamin D3 economically, so it can be, we're not fishing out the sea or harvesting all the animals to get sufficient amounts for 7 billion people.
Absolutely. Listen, James, this has been tremendous. It has been a tremendous honor to have you with us. I think that not only myself, I have learned a really valuable thing here. I'm going to take forward in my research. I think it has been of value to the listeners with some of the questions about vitamin D. I really appreciate this. Thank you very, very much for the work that you are doing, James. And we look forward to hearing from you in the future.
Thank you, Philip. And it was quite an honor to be interviewed by you. Thank you.
Wonderful. Have a great day.
You too.